Thank you to the organizers for inviting me. And again, full disclosure, I work for a small company, Ocell, in Mountain View, California, that's developing live biotherapeutic products. And I'm a guest at NIH on a CRADA. All right, I have to remind you that among primates, the human vaginal microbiome is unique in that it's often lactobacillus dominant. And whether you look at a gram stain or this bacterial communities plot with a number of different women represented in these bars, you can see that there's a single species that's dominant, which is a lactobacillus. Now, a common condition in women that shifts the microbiome from low diversity to high diversity is called bacterial vaginosis as a clinical diagnosis. And you can see here a dysbiosis in the gram stain by seeing, again, many different species of bacteria. And in the bacterial communities plot, you see this high diversity microbiome with Synethias and Gardnerellas and Previtellas and other organisms present. And I want to add that this microbiome makes women more susceptible, especially young women, more susceptible to HIV infection. In humans, humans have lower vaginal pH than primates, around 3.6 to 4.5 when lactobacillus dominant, greater than 4.5 when they have the condition called BV, and about 5.5 to 8 in non-human primates. So we've been using non-human primate models to test live biotherapeutic products. And I'd like to make a distinction. Live biotherapeutic products are different than probiotics. LBPs are developed as drugs to treat, mitigate, or prevent disease in humans, and probiotics cannot make that claim. We used human vaginal lactobacillus in macaque colonization studies. We've used Lactobacillus crispatus, which is one of the most common human vaginal lactobacilli, but most of the studies I'll talk about today are with a recombinant Lactobacillus jenseni, which expresses an HIV entry inhibitor, cyanovirin, which I won't go into in great detail today, but it's being developed as a recombinant live biotherapeutic for HIV prevention. Now, I think Jeff Lifson covered these models very well this morning. We've used all three models, rhesus, pigtails, and cynos. And he talked a lot about this again, but I'd like to point out that these animals do have similarities to humans in that they have overt menses that last anywhere from 1 to 5 days, and they have about a 28-day cycle. So again, very similar to humans. These animals mature at about between 3 and 4 years of age, and for rhesus, they can be fall-winter breeders when they're housed outdoors, but in captivity they have no distinct breeding season, whereas pigtails have no distinct breeding season, and again in captivity, cynos have no distinct breeding season. However, the macaques have a very different microbiome, which is more complex, so more similar to women with BV. This is a gram stain that we perform, so you can see multiple morphotypes. And in these two rhesus macaques shown here in a study by Greg Spear at University of Chicago, Rush University in Chicago, you can see that Synethia and Mobiluncus and Previtella are the predominant species, and of course other unidentified bacteria. And then lactobacillus makes up a very small percentage of the microbiome for rhesus. Same is true in pigtails. This is a gram stain we did a while back with a lot of different bacteria represented. And in this bacterial communities plot that followed animals longitudinally, again from Greg Spear's group, you can see that again, reading from the bottom, Synethia, Fusobacterium, Previtella, Porphyromonas are the major species, and lactobacillus, again, are present in many animals but represent a small percentage of their microbiome. And finally, in cynos, we see, again, these are 5 different cynos, and this is a composite of the 5. And again, Synethia, Porphyromonas, Previtella, and Fusobacteria make up the majority of the microbiome, with lactobacillus here in this one animal being a very small percentage. So glycogen is a key factor for lactobacillus. It's a key nutrient for lactobacillus. And humans have higher glycogen levels than non-human primates. Here in this diagram, we can see that women with low Nugent score, and Nugent score is just an assessment of the vaginal microbiome using a gram stain. So these women would be lactobacillus dominant, and they have about 2 micrograms per microgram of protein of glycogen. Women with bacterial vaginosis with Nugent scores that are higher have about a 5-fold lower level. Rhesus macaques have about 50-fold lower than humans, and pigtails had very negligible amounts, although there was variation seen in pigtails over the menstrual cycle. So we have used, again, colonization of the macaque with a human vaginal lactobacillus ginsenii. And so generally, we treated macaques with antibiotics, azithromycin, for about 5 days to clear out some of the microbiome. And we colonized them for 5 consecutive days with an LBP. And we started with live lactobacillus, and later on we used lyophilized powder, tablets, and we got colonization with all of these. And generally, we took swabs and cervical vaginal lavage to assess colonization. We looked at all 3 models, and in our hands, the Rhesus macaque gave us the most long-term colonization. We had animals that were often colonized for a couple of months with our lactobacillus, and at fairly high levels by culture and by qPCR between 10 to the 5th and 10 to the 8th. Pigtails, we also saw colonization. This is a study we did with Dorothy. There was a little bit shorter term. We saw out to 14 days post-inoculation, again at fairly high levels. Oh, I want to note that both of the Rhesus and the pigtail, we could see recombinant protein being expressed from our lactobacillus. So we know that they were colonized to the extent that the bacteria were happy in producing protein. In cynos, we saw very transient colonization that lasted about 7 days, but dropped to about 10 to the 3rd, which is about our limit of detection by 1 week. So we then decided, based on this data, that the Rhesus macaque would be the best model for us to use. So in this study, we looked at the relative abundance of lactobacillus in macaques that were treated with an antibiotic and then vaginally dosed with tablets that contained lactobacillus, or placebo tablets for 5 days and then once a week over 11 weeks. And we could see a significant increase in the lactobacillus in the treated animals over time. Concomitant with that increase, we also saw the increase in lactobacillus was accompanied by decreases in Proteus, which is a gamma proteobacteria, Bacteroides, and Veillonella, which is a firmicute. So specific genera were actually being suppressed by the lactobacillus. And in this study, when we looked at 16S rRNA in this longitudinal study, we could see that animals that were dosed with a placebo, we can see some transient lactobacillus in these animals, but in general, we didn't see lactobacillus as the dominant species. And yet, in macaques treated with antibiotics and dosed with this lactobacillus LBP, we could shift the microbiome significantly toward lactobacillus dominant, very similar to humans. We did see shifts in the microbiome during menses in the animals. You can see a major shift here and here. This is also commonly seen in humans. In women during menses, often their lactobacillus levels will drop. So all macaque species have highly diverse vaginal microbiomes. Rhesus macaques dosed with our lactobacillus showed sustained colonization, and we could shift the vaginal microbiome toward lactobacillus dominant. And interestingly, I didn't have time to talk about this, but macaques made no antibody response to the lactobacillus, so they considered it a normal part of their composition, their microbiome composition. We went on to show that our lactobacillus recombinant, when it colonized the macaques, we could see protein expressed in cervical vaginal lavage 1 day after, just as early as 1 day after inoculation and as late as 6 weeks, and this was without re-inoculating the bacteria. And we can see that the protein was located on the vaginal mucosal surface. This is the surface and the lumen in the vagina, and this is our anti-cyanovirin, our entry inhibitor protein. And macaques that were colonized with our Lactobacillus ginsengii producing cyanovira were protected from a Shiv challenge. And so I believe that as the field in HIV hasn't really talked a lot about the microbiome, but microbiome modulation is moving forward in many other fields, and I don't know whether it will move forward in the HIV field, but certainly in women with bacterial vaginosis, to shift the microbiome toward lactobacillus to a lesser inflammatory microbiome could be useful in future HIV studies. So in conclusion, all three models could be used to mimic the human vaginal microbiome. Certainly during bacterial vaginosis, there was a complex, diverse flora or microbiome in all macaques. The Rhesus macaques in our hands was the best model to test our live biotherapeutic lactobacillus, and Rhesus macaques dosed with recombinant lactobacillus expressing cyanovirin could be protected from Shiv challenge. And I accidentally deleted my acknowledgement slide, but I'd like to acknowledge I didn't mean to do that. And I tried to, I tried to copy it and bring it here, and then I didn't have the right acknowledgement slide, so I want to acknowledge my collaborators at OCEL, Jacques Ravel and Greg Spear, who shared some data with me, and of course, NIH, who's funded some of this work. So thank you.
Okay, we can take some questions. Can you make the lactobacillus less dependent on the glycogen to use in the monkey model?
Clearly, it's not totally dependent on glycogen, since both rhesus and pigtails, which have low levels, could colonize. Right. You could put other carbon sources in when you apply the live biotherapeutic, because since it would be reapplied. Christine?
Yeah, I have a question. I think it's really fascinating that we can change it for a relatively long period of time. But there are now interesting data emerging, especially by Jacques Ravel too, that there's clearly a racial difference in the human vaginal biome. And in Zambia, I know, like our collaborators, big thing that Black women, the vaginal microbiome is less lactobacillus dominant. So actually, in the African population, even in HIV uninfected, you see a lot more of the non-lactobacilli dominated vaginal flora. And that's something you see in Black women in the US too. So I think if we want to address the problem of susceptibility to HIV infection, that would be a major undertaking if you want to change the vaginal microbiome of a whole race. And I wonder why that is the case, and if that has some other advantages in certain regions. And it's interesting that the vaginal biome in African women is actually really a lot more similar to the vaginal biome in Black women in the US, which really means that there is a race component to it.
So there may be a race component, genetic components, vaginal practice components, multiplicity of sexual partners. Certainly Black women and African women can have lactobacillus and can be lactobacillus dominant. When they do have bacterial vaginosis or diverse, they have high numbers of inflammatory cytokines. So it's not a good thing for them to not be lactobacillus dominant. We are starting a study with a fresh cohort to introduce Lactobacillus crispatus into those women and to try to shift them toward lactobacillus dominance to reduce their inflammation.
Hi, Laurel. Jason Brenchley, NIAID. A quick question. Is it possible that the prolonged colonization in Rhesus compared to pigtails or cynos is at least partially attributed to the more I guess, non-predictable menses that Rhesus have relative to the pig. So if you look longitudinally, maybe one Rhesus doesn't menses for 3 months, and then when it does, it's no longer lactobacillus dominant. So if you look pre and post menses, and then do the comparison between Rhesus, pigtails, and cynos, is it still true that Rhesus colonize for longer periods of time?
Right. So the Rhesus we had were housed in 12-hour light cycles, and they did menstruate during the study. We still got colonization in those animals, but that certainly could be a contributing factor.
Hi, Alex Ballison, Reagan Institute. I'm just wondering, did you see lower levels of inflammation in your monkeys that were colonized with lactobacillus? And is it that, or is it the cyan of urine?
So certainly in women, they've shown that cytokine levels, when women go from a BV state to lactobacillus, their cytokine levels go down. In the macaques, we looked at cytokines during our study, and they really didn't change. So I think the macaque diverse microbiome is less inflammatory to begin with, because that's what they've adapted to. So that's what